So, hello everyone. Thank you for joining us today and welcome to the 8th lecture of the current topic Inheritance Science series of E-RIHS Academy. These series are organized by emerging professionals within the frame of E-RIHS project and the European Research Infrastructure for Cultural Heritage. My name is Moira Bertaza. I'm the heritage scientist at the Historic Royal Palaces. And today I'm going to moderate a lecture focused on X-ray imaging in cultural heritage studies. The recording of this lecture will be available later on the E-RIHS YouTube channel. Furthermore, at the end of the lecture, you will receive a survey. So please take a bit of your time to let us know your opinion about the series. And please be aware that the attendance certificate of the lecture will be sent only after the survey submission. You can ask questions using the Q&A function, and they will be asked to the speaker at the end of the lecture. In case you will experience any technical issues, please let us know through the chat. So our speaker today is Dr. Daniel O'Flynn. He is an X-ray imaging scientist in the British Museum's Department of Scientific Research. He is responsible for the operation of large-scale X-radiography and CT facilities housed beneath the museum. His research applies advances in imaging to gain insight into ancient cultures through the manufacture and the use of objects and the lives of people and fauna. Today, we'll present a lecture titled X-ray Imaging in Cultural Heritage, the Past to the Present. Dr. O'Flynn, thank you for being here today, and please, the floor is yours.
Thanks very much, Moira, and thanks for the invitation. And I'm happy to see so many people here as well today. So I'm just going to start sharing my slides now. And hopefully you can see those.
Yeah.
So as Moira said, my talk is about X-ray imaging I'm going to start at the very beginning and then go to some more recent uses of X-ray imaging. So the X-ray was discovered in 1895 by Wilhelm Röntgen. He published a paper in the same year, which was translated into English in 1896, called On a New Kind of Rays. And figure 1 of the paper was an X-ray image of his wife Bertha's left hand with her wedding ring present. And it was very quickly understood the potential for X-rays to study under the surface of people or animals without disturbing the surface. So within a year of this publication, but of course, Röntgen won the Nobel Prize, the first ever Nobel Prize in physics for his discovery. But within a year of this discovery, there were radiographs taken in cultural heritage context. So these two images that I show on the right-hand side here, the image of in the center is of the knees of a child's mummy, and on the right is a cat mummy, and they were also taken in 1896. And then just a few years later, there was another example in the British Museum, which I'll go to next. But just to say, on a terminology note, these are what we call radiographs, these images, and I'll talk about the distinction between X-radiographs and X-ray CT scanning later. But a radiograph is a two-dimensional image in which every layer under the surface of a mummy or of an object is superimposed. So a few years later, in 1899, an X-ray tube was brought to the British Museum, and it was used to image a baboon mummy. And the resultant image was published in a book on the zoology of Egypt in 1902. And the baboon was identified as a juvenile Hamadryas baboon. And we can see on the X-ray image here, we can see that the tail of the baboon is wrapped around its lower legs. We can see that the spine has collapsed below the shoulder blades. We can also see, quite interestingly, if I just zoom in on the skull here, the canines of the baboon are still present. This is quite an interesting thing to see because they were often removed in captive baboons in ancient Egypt, presumably to reduce the risk of injury to humans from those teeth. So these kinds of X-ray imaging, like radiographs, are still being carried out to this day in museums. This is a study which came out last year on ancient cancer, and this is a collaboration I was involved in, led by my colleagues in the British Museum's Department of Egypt and Sudan. And this was an X-ray and visual examination of the skeletal remains of a probable male individual, thought to be over 50 years old at their time of death, discovered in a cemetery in Kerma in the north of present-day Sudan. And only 30% of the skeleton is now preserved. And using a combination of visual inspection and X-radiography to look below the surface, we were able to detect indications of cancer. So the close-up image on the right here is of a lower rib, and the dark regions you can see with bright outlines inside the rib are lesions. So these are areas of bone loss and regrowth inside the rib. So these types of response were found in several different bones in the body, which suggests a metastatic cancer-induced bone response. So X-ray imaging is still very useful today in museum context. Just to give a little bit of a background onto how X-rays actually work, I wanted to talk very briefly about X-ray attenuation, and this is the process by which the intensity of an X-ray beam is reduced as it passes through an object. So what we have here in the figure is an X-ray beam with an intensity of I0 passing through a material with a thickness of X. And this material will have a property called an attenuation coefficient. And this attenuation coefficient is related to its density and the different atoms inside the material. So what happens when the X-rays pass through this material is that there is an exponential decay of the intensity with increasing object thickness and also with increasing attenuation coefficient. And the physical process which causes attenuation, there are two processes. There's photoelectric absorption and Compton scattering. And it's photoelectric absorption. So when the X-rays are absorbed by the atoms inside the material, this is the process which we typically exploit when we take X-ray images. And then X-rays interact with electrons surrounding atoms. And the higher the atomic number, the higher the number of electrons, and thus generally more X-ray attenuation. This is why X-rays don't readily pass through lead, for example, when compared with carbon. So higher energy X-rays are more likely to pass through objects without being attenuated. And that's something that we can sometimes use when we're taking images in a practical sense. So what happens when you have two different types of material with two different thicknesses in the beam path? Then you can sum together their thicknesses and their attenuation coefficients in the exponential decay formula here, and you'll still be able to derive the intensity. But how do you separate them out afterwards when you take an X-radiograph and they all overlap? You can't separate them. So a new method was needed to be able to separate complicated three-dimensional structures. So in the early 1970s, Hounsfield and Cormack developed a technique independently called X-ray tomography. And this means to take X-ray images in slices or in cross sections. And X-ray tomography is also known as computed tomography, aka CT scanning or CAT scanning even. So there's lots of different ways of naming the same technique. And what this technique does, if you can see the X-radiograph of the chest on the bottom left here, you can see that it's quite hard to make out organs, for example, on an image like this, because everything's overlapped into a 2D plane. With the image in the bottom middle, that's an X-ray CT sliced image through the abdomen. So you can see the vertebra at the bottom of the image there and the ribs surrounding the outside. And you can see two kidneys there as well in the image. So we get much more detail on complicated 3D structures like the inside of the human body with CT scanners. The first commercial scanner was made in 1971, and then 8 years later, they both were awarded the Nobel Prize for their discovery. And there are many different ways of doing CT scanning. I'm going to talk to you about them now. So on the left-hand side of the image, of the slide here, is my lab that I'm responsible for running in the British Museum. And on the right-hand side is what's called a micro CT scanner. A micro CT scanner is another type of CT scanning where you can get very high resolution by using an X-ray tube with a very small focal spot. And it means you can magnify small details to image down to the micron scale. And They are quite different in terms of their scale, but they both contain the same elements, which are an X-ray tube, which shines a beam of X-rays towards an imaging detector, and you place an object in between the two and you take a picture of it. And in order to do a CT scan, you need to rotate your object as well. So they're both equipped with rotation stages. And you may be more familiar with an X-ray CT scanner that you would see in a hospital, and they typically look like this. And again, it's the same principle, but it's done in a different way. So here, what we have is an X-ray tube and a detector inside this donut-shaped structure, and they rotate around the patient in the case of a medical CT scanner. But they're all three different types of setup here are doing the same thing, and they're taking an X-ray image of an object from multiple different angles, and then using that to reconstruct a three-dimensional picture of its inside. So why might we use X-ray CT in heritage science? A very brief overview. We often want to know, when we're looking at objects, we want to know how they were made and what their current condition is. And this can help us to understand ancient manufacturing processes, for example. We sometimes want to know if there's something hidden inside. That's when you have a box or something hidden under wrappings or in under a layer of corrosion or soil, for example. We're able to look inside using X-rays. We can also use X-rays to further understand the lives of people and animals. And I've already spoken about ancient cancer, and I'll show some more on the next slide. And the key with X-ray imaging is that it's non-invasive. And of course, X-ray imaging produces very visually striking results. So these have been used to great effect in exhibitions in museums. And I'm just showing a few examples here which are related to the British Museum in some way or another. So there was an exhibition called Ancient Lives, New Discoveries, which was about 9 years ago in the British Museum. And 8 mummies were scanned in a medical CT scanner, and the results were shown in the exhibition as digital displays. And on the image and on the left-hand side here, there is a segmentation of the CT scan, which is highlighting different amulets inside the mummy of Tumut here, including representations of a scarab, a falcon, and a winged goddess, and they were all able to be revealed from the CT scan without disturbing the wrappings. And the image at the bottom is of a crocodile mummy, which is nearly 4 meters long. And then this crocodile was also scanned in a medical scanner, but in this case, a veterinary scanner, which normally was used to scan horses. And they scanned this 4 meter long crocodile inside the CT scanner. And again, this was on display in the British Museum as a digital display alongside the mummy of the crocodile. And there was some evidence of the mummification practice here. So the red highlighted material inside the crocodile is packing material, which was placed inside the crocodile when some of the organs were removed. And the bits that are in like a light orangey brown color, they're actually the bones of a cow or an ox. And so apparently this was a captive animal which was fed the best cuts of meat prior to its sacrifice. And in the exhibition, these bones were actually 3D printed from the CT scan. So you could physically see what's inside the crocodile. And again, this is all non-invasive. And a recent study, which I was involved in collaboration with the National Museum of Scotland, was to look at the Galloway hoard, which was a Viking Age hoard, which was discovered in Scotland a few years ago. And with X-ray CT, we were able to look underneath the textile wrappings still stuck to the surface of this vessel. So there's a photograph of the vessel on the left, and on the right-hand side is the result of the CT scan. And the CT scan revealed decorations on the surface of this vessel, which changed the narrative about its origin. And now it's thought to have originated in Central Asia and somehow made its way all the way to Scotland before it was buried. And again, a 3D print of this scan was used in the exhibition at the National Museum of Scotland a couple of years ago. So I want to now talk about the procedure for taking an X-ray CT scan. So how do we practically do it in the lab? So I'm using the example of an Islamic ewer, which I CT scanned previously, and in the British Museum X-ray lab. And I'll show you the step-by-step process of how we did the CT scan. So the viewer here on the photograph on the right is set up in the lab, and that the object you can see to the right-hand side is the X-ray tube. So when we take a single X-ray image of the vessel, it looks like the image there where it says radiographic projections. So we don't just take one image, we take hundreds of images, sometimes thousands of images, and each image is rotated slightly. So we end up with a series of images, which I'm playing as a movie here. And we do that through a full 360-degree rotation of the object. And once we've done that, we have a lot of information which we can use to reconstruct the slice-by-slice data of the vessel. And we can view that in two different ways. We can view that as a CT slice, which is like a slice of bread, if you like a slice right through the middle of the vessel, which shows us all of the internal features. We can also view it as a volume render, which is the video that's playing on the right-hand side. And that's a false color image of a three-dimensional version of the vessel entirely from the CT scan. And that I skipped a step a little bit there, so I'm going to try and explain it in a bit more detail on the next slide. So how do we go from the radiographic projections to the CT slice? So I'm going to use a 2D example because it's a little bit more simple to explain. So this is an example object of three shapes with different density, and the more dense objects are brighter in this case. So the circle is the most dense object and the triangle is the least dense. If we shine a beam of X-rays through this object from the bottom to the top, we see a signal as shown here at the top of the image. And this signal is a measure of the X-ray attenuation. And you can see here the peaks that are showing in the shape of the object. So we can see a triangle and a square and a hump, which is due to the circular object. Now, if we instead were to rotate the X-ray beam so that it was coming from the left-hand side of the object, then we would see a different signal entirely. Now, from the point of view of this 90 degrees rotated version, all of the three shapes overlap and we see something very different. So that immediately shows us that we're learning something new about the object by taking X-rays from different angles. So how do we use this to reconstruct our slice? So let's start with a blank space for our reconstructed slice and take our first signal. And this process is called filtered back projection. So I'm going to show you the back projection part first. So we take our signal and we add it along every position back towards its source and effectively smearing it across the whole slice. And then we see something like this, where we can see our triangle is this part of the left-hand side. Then we have our square in the middle and then our circle. And it doesn't look much like what we're expecting so far. And now if we add our signal from 90 degrees, and again, we're going to back project, but we're going in a different direction now because we're 90 degrees rotated. We're going to add that signal on. Then we see something that looks like this. So that's only two projections. And I said before that we sometimes use hundreds or thousands of projections. So if we keep increasing the number of projections that we take, then we start to see the shapes appearing gradually until we get something that looks like this. So that's what happens if you do just back projection. But what we actually need to do is apply a filter to these signals before we do the back projection part. And when we apply the filter and do the back projection, then we see something that looks like this, which is our reconstructed slice. And it's never perfect, but this is a pretty good representation of what we started with. And that's the process by which we use to get tomographic slices. So, of course, like any experimental procedure, tomography has many challenges to it. One of the main ones from a heritage perspective is the experimental setup itself. So the ideal shape for a CT scan is a cylinder. And that's because when you rotate an object, you need to keep it inside the field of view of your imaging detector through its full rotation. And as you rotate a cylinder, it stays the same. So you don't have variations in width. Of course, in cultural heritage, we don't have cylinders. Most of the time, we have all sorts of different object shapes and sizes. We typically, for a CT scan, need to mount it upright so that it moves as minimally as possible. Its width varies as minimally as possible throughout a rotation. So the best way to do that is typically to mount something upright. That's not always possible, or you need to have a lot of thought put into how you mount your objects in order to take an effective CT scan. And of course, you need to consider delicate material in this. Yeah, I mentioned the field of view briefly before. So this is related to the size of your detector and your system geometry. So how close can you get your object to your X-ray tube, to your detector? How much information are you trying to get? If you need to fit your entire object in your field of view and your object is bigger than your detector, then you instantly have a problem, which you need to overcome with some kind of experimental adaptation. The spatial resolution of CT scanning is very high if you have the right system, and that means you need a very small focal spot on your X-ray tube and also relatively small pixel size on your detector. And any variation in that generally means that you don't have such a high resolution. In a medical CT scanner, the resolution is actually quite low because the most important thing with such a scan is to keep the dose to the patient as low as possible. So in a laboratory environment, you can technically get higher resolution, but at the cost of higher dose. X-ray attenuation, which we covered earlier, can be a very big problem if you have something very dense, like a piece of metal inside your object. And the image on the right-hand side here is of a vertebra, of a CT slice of a vertebra with a metal implant. And you can see the very bright region is where the metal implant is. And the radial streaks that are coming from that are CT reconstruction artifacts. So they're not real, they're not present in the patient, but they are a side effect of the X-ray attenuation from the heavy element in the implant. The radiation dose is a very important thing to consider. We often refer to X-rays as non-destructive, but they're not truly non-destructive. Very high radiation doses can cause damage to objects or to remains of humans or animals. So, for example, X-rays could cause discoloration of materials, and at very high doses, they could cause damage to DNA structures in humans or animal remains. So there's a lot of research to be done to further understand these effects. And we definitely don't have all the answers right now, but I think we definitely need to exercise caution when using X-rays with heritage material. And then the last thing I wanted to cover here as a challenge, it's a very important one, is the data size. So when we take a CT scan, we generate tens or sometimes hundreds of gigabytes of data. So that needs consideration in terms of how you store it and also computing power to actually access and manipulate the data. So I wanted to show a few examples from the cultural heritage world of X-ray CT and why we use it and why it's useful. So this is a clay tablet from ancient Mesopotamia. It's about 4,000 years old, and this is actually a tablet which is inside an envelope, and they're both made of clay, the tablet and the envelope. So, and the writing on the surface of the envelope, which you can see here, is a script called cuneiform, which is the earliest known form of human writing. And you can just see there's some damage to the top left corner of the envelope here, and you can see the tablet inside poking out. So what we have is clay and little indentations in the surface, which are the writing. And what we want to be able to do here is to read the writing on the inside. And obviously we don't want to damage the object physically, but with the attenuation properties of the clay, it means that when you take a radiograph, you see something that looks like this. And that's great if your question is there a tablet inside? And you can see the outline of it, and you can see the air void around it. But that's not very useful if you actually want to read the writing on the inside. So if you take a CT scan of the same tablet, and you end up with a series of slices. I'm just highlighting two here. So the blue region and the yellow region are two different heights, and taking cross-sectional slices through the tablet and the envelope. And if we look at the top image first, you can see I've indicated with arrows where the envelope is surrounding the tablet on the inside. And if we go down to a different depth, we can also see the same thing. And in this case, the tablet is fully surrounded by the envelope because it's the region where it's not damaged. And something I want to highlight on these slices, and I'm highlighting them with the arrows here, are these small triangular voids that you can see. And these triangular voids are where the writing is present on the inside. So, and we see this in a different way. So we'll go now to a volume rendering of the same tablet. So this is again on our CT scan on the right-hand side. It's just a different way of looking at the same data. And it's not just the surface, but it's every layer under the surface. So we can digitally peel back the layers of the envelope and go inside the tablet. And what you can see here, these triangles that you can see inside, this is the writing on the inside of the tablet, which has not been seen in 4,000 years since the tablet was sealed in an envelope. So we can then take that data and we can process it, and then we end up with something that looks like this. And I took this to our curator in the Middle East department, Jonathan Taylor, who is one of the few people who can read cuneiform, and he provided the translation, which is on the left-hand side here. And this tablet describes a transaction, or the movement of a wool, a very large quantity of wool from a big city called Girsu, which is in present-day Iraq, to a small village called Gaka. And it has the date of the transaction, the names of the people involved in this, as well as the name of the person who sealed the envelope. So it's an administrative document which has been perfectly preserved in this envelope, and now I'm covered with X-rays. So not only can we use CT to look through clay envelopes, we can also use it in the case of tablets where, due to the conditions in which they were kept, they've been encrusted with salt on the surface. And this is probably due to the high salinity of the clay in which the tablets were made from. And when they were kept in different conditions, salt penetrated the surface from the inside and crystallized on the surface. And in some cases, the salt entirely obscures the writing on the surface. So the quote at the bottom of the slide here is from the person who set up the department in which I work, the Department of Scientific Research in the British Museum over 100 years ago. And he refers to this issue of salt on cuneiform tablets. And so on the left-hand side of the image here is one side of the tablet in which we can see the text is fine. On the other side, the salt is completely obscuring the writing. And on the right-hand side of the slide is a slice through this tablet. And you can see the very bright region on the lower part of this slice. That's the salt, and it's more dense than the clay. And that's why it appears bright on this image. And again, with the arrows, I'm highlighting these triangular indentations, which are the writing. So what we can do here is we can digitally segment or isolate this salt, and then we can delete it, and then we can reveal the writing on the inside. So again, an entirely non-destructive way of uncovering material which was waiting for the right technology to come along. So there was a lot of foresight from people to not try and remove the salt and damage the tablet. And one more example I want to show you from the British Museum is in the manufacture of ancient Egyptian mummy masks. And these masks were made from a material called cartonnage, and then they were painted and gilded afterwards. And this was a study that we published a couple of years ago. And I just want to show you this one mask because it has some very interesting features on the surface and on the inside. So this is on the left-hand side is our image of the photograph of the mask in the X-ray imaging lab. And in the center is a volume render of the CT scan of the mask. And on the right-hand side is a tomographic slice in side view right down the middle. So you can see the nose protruding on the right-hand side of the slice. So something I can point out is that the textile layer, which is in the lighted gray there, is much thicker in the region of the nose. And that's because extra layers of linen were applied as a reinforcement in this area because it's the fragile part of the mask. And the bright outline on the right-hand side of the slice is a material called gesso, which is a binding material mixed with chalk, gypsum, or pigment. And then on top of this layer, the mask was painted and it was gilded. And the gilding was done with very fine pieces of gold leaf, which were applied to the surface of the mask. And there were overlaps when this gold was applied to the surface. But the people who made the mask can disguise that very well by burnishing, which essentially means rubbing the gold until the joins disappear. But if we zoom in on the CT scan, we can see where the arrows are indicating the regions of the overlap of all these different pieces of gold on the surface of the mask. So as well as the outside layer of the mask, we could also obtain an impression of the inside of the mask as a 3D. volume. And this isn't a human face, this is a mold that was used to create the mask. But we're hoping that with enough study of different types of mask, we might start to see some similarities in the molds used to learn more about their provenance. And I'm just going to briefly touch on this because it's a study which is ongoing, but this is some work I'm currently involved in on ancient Egyptian animal mummies. And we are aiming to understand mummification practices and cause of death of the animal and maybe the conditions in which the animals lived through X-ray CT of their remains. And one of the challenges here is that mummies of animals were often in different types of containers. So I just thought I'd highlight a cat mummy on the left-hand side, which is wrapped. And then next to that is a wooden statue of Sekhmet with a human with a lion head, inside which is a cat mummy. And then there in the center image is a ceramic vessel with an ibis mummy inside. And on the right-hand side are two metal boxes, which are thought to contain the remains of lizards. And metal is very hard for X-rays to see through. So they're all presenting different challenges for our work. And on the bottom, there is a volume render of a very small crocodile mummy, just to show some of our preliminary results here. So on the subject of crocodile mummies, I wanted to touch on some research from another group, which is fantastic. They used a synchrotron, which is a central facility which generates X-rays with very high flux and lots of properties which make them useful for X-ray imaging. And in this study, a crocodile mummy was studied using a technique called propagation phase contrast microtomography. And this technique is very sensitive to small details. It can give much better contrast than the traditional absorption-based tomography we've been talking about so far. And with this study, a wrapped crocodile mummy was scanned, and the stomach contents are shown here. And a lot was found in the stomach, including insects, fish, soft-shell eggs, and a complete rodent, which is part G of the figure there on the right-hand side. So based on the variety of material inside the stomach of this crocodile, the authors presumed that this was a wild crocodile which was hunted rather than a captive crocodile, which would have been fed choice bits of meat like the crocodile I showed you earlier on in the presentation. And they also found the likely cause of death in a large skull fracture on the crocodile, which was later filled with a resinous material. And another quite novel use of imaging, which I'm only going to touch on here, is neutron imaging. And neutrons you can do radiography and you can do tomography just like you can with X-rays. Neutrons are a complementary, non-invasive imaging technique to X-rays. They're much better at looking inside metal objects, neutrons. They're interestingly very sensitive to hydrogen and not very attenuated by lead, which means that they're really good at looking at organic material inside metal objects. So in this study by Lehman and Hartman, I'm showing here, X-rays provide detail in the bronze sculpture in the central image, but neutrons reveal an upright wooden stick in the center of the sculpture and a mass of plants inside the base, something that you would never notice on an X-ray image. So it's a very powerful complementary method to X-ray imaging. So that brings me to the end of what I wanted to talk about. I'll leave this slide up briefly if people want to take a screenshot of some further reading. There are, just looking at the articles here, there's a really nice introduction to the process of X-ray CT, which came out a couple of years ago, and some discussions on how best to take CT scans and the kinds of artifacts you might encounter, and a user guideline, as well as some articles highlighting really interesting examples of X-ray CT when applied to cultural heritage material. And then the bottom article here is the neutron imaging work I just spoke about. And I think that brings me to the end of my talk. Thank you very much.
Thank you so much, Dan. It's always amazing to hear about your research. And but before going through all the questions, if you can show yes, I would like to invite all the attendees to the next lecture on the 18th of May, where Dr. Michela Botticelli from the University of Glasgow we'll talk about the ethical of sampling. So furthermore, in preparation of this lecture, you might be pleased to reply to another interesting survey about the ethical consideration and practice of cultural heritage sampling. And here you have the link, and I think you have also the link in the chat right now. So as usual, the next lecture will be at 3 p.m. Rome time.